Good afternoon, and thanks for tuning in to my presentation. I'll start by providing a very brief introduction to our company and the services we provide. Biocytogen was founded in 2008 in Worcester, Massachusetts. We initially focused on genetic engineering, where we've developed proprietary methods that improve efficiency even for very large insertions. We've been able to supply over 3,000 custom animal models for both academic and industrial clients worldwide. Harnessing these technologies, our company has since created a vast array of humanized animal models to further support the research endeavors of our clients. After growing into one of the largest animal model suppliers in China, we opened our branch in the greater Boston area in 2018 to expand our services. For antibody drug discovery, Biocytogen has specially designed mouse models to facilitate the discovery process. Using an advanced chromosome engineering technology, we recently introduced our fully human antibody mouse, RenMab, which contains the entire human immunoglobulin variable domain, including the surrounding non-coding regulatory regions in C2. The use of this mouse and its related platforms, RenLite and RenMice, its platform, which I'll briefly mention a little bit later, are powerful models for fully human antibody discovery applications. After antibody hits are generated to your target, we offer various models for later stages of antibody drug discovery, including humanized mouse for immuno-oncology targets, which I'll be focusing on today, cytokines and cytokine receptor humanized mouse models, and immune system humanized mouse models using immunodeficient mice. For the rest of today's presentation, I'll discuss some of the general challenges that have been faced in bringing some immunotherapies to clinic and how humanized mice can help to bridge this gap. I'll provide specific case studies in the context of 3 different experimental models. Finally, I'll summarize how these models can be seamlessly integrated into our antibody drug discovery platforms that can ultimately help you streamline your therapeutic drug discovery application. So why do we need humanized mice? Well, many new therapeutic drug candidates struggle to cross the bridge from basic science to late-stage clinical trials and get trapped in the so-called valley of death. One way to improve the translation is to improve the methods used to assess the clinical utility of new drugs earlier in the discovery process to reduce attrition rate. For antibody therapeutics that might have poor cross-reactivity across species, it can be challenging to perform and obtain meaningful results in initial test studies and animal models. So how do we do this? At Biocytogen, we've placed a lot of emphasis on improving the mouse models used during the discovery process by using various flavors of humanized mice, including those with a reconstituted human immune system or humanized targets via genetic engineering, which is most relevant to today's talk. We hope to improve clinical translatability of new therapeutics. At Biocytogen, we offer various humanized animal and cell models to support preclinical research. So for the purposes of immuno-oncology drug discovery, I'm going to focus on the humanized immune checkpoint mice, but we offer many other models and pharmacology services using these models in our toolbox, including immune system reconstitution in immunodeficient mice and humanized mice for cytokines and receptors. We're also able to humanize tumor cell lines for some of our efficacy studies, which I will introduce a little bit later in today's presentation. We've worked hard to generate, validate, and offer multiple humanized immune checkpoint mice and many multigenic immune checkpoint humanized mice. These are simply some examples that include some I'll be describing today, but for a comprehensive list, please check our catalog, which has been made available by our team, or head over to our website and search for your model of choice. So some of the advantages of using humanized mice for evaluating immuno-oncology drug efficacy is that the animal has an intact immune system as opposed to an immunodeficient model engrafted with human PBMCs, which might not develop properly. Because these studies are in vivo, they allow for mechanistic studies to confirm that therapeutic candidates are performing their function as intended. Because they're mice, the studies are lower cost and it is much more feasible to study larger cohorts. The main disadvantage is that the mouse and human immune system are not completely identical, so in some cases, disease may not be completely recapitulated. For proper receptor signaling, we may need to create doubly humanized mice for the receptor to interact properly with its ligand and maintain function. However, when this is the case, it allows for testing of novel combination therapies. The first model I'd like to focus on is our humanized CD3 mice, which now comes in two flavors. Our first generation is mice expressing human CD3E. As we all know, CD3 is a popular immuno-oncology target because CD3 activation plays such an important role in T-cell activation. Many bispecific antibodies have been engineered to target CD3 in addition to tumor surface antigens in order to directly juxtapose the T-cell, which is shown here in blue, with the tumor cell, which is shown here in green. In this way, engagement of the CD3 complex with the antibody triggers a T-cell response, ideally resulting in tumor cell killing and/or T-cell activation and cytokine release. With the number of anti-CD3 drug candidates steadily increasing, there remains some challenges in identifying those that are the most safe and effective in preclinical studies. This is because therapeutics specific to human CD3 cannot effectively activate murine effector T cells due to the low sequence homology between mouse and human CD3. Thus, humanized CD3 models are very much needed to test novel CD3 specific therapeutics in vivo. In the next few slides, I'm going to highlight the detailed design and characterization studies we performed for this particular model to ensure normal immune responses and to give you a flavor for how we generate and validate our models at Biocytogen. So in this model, the extracellular domain of human CD3E, which is shown here in pink, was inserted into the murine CD3E locus to replace the murine extracellular domain. mRNA expression demonstrates expression of human CD3E by RT-PCR, which is shown in the pink box. And importantly, expression of human CD3 but not mouse CD3 is detected on the surface of homozygous CD3E T cells by flow cytometry. Previous attempts to generate humanized CD3E mice have met significant challenges, with early attempts resulting in completely defective T cell development and some others reporting altered immune function. So here in our model, gross assessment of the lymphoid organs from these mice indicate that while there is a reduction in the overall appearance and weight of the thymus, there is not in the spleen. The thymic weight reduction appears to be due in part to a reduction in thymocyte number in the humanized CD3E mice. However, despite this reduction, humanized CD3E mice exhibit normal leukocyte and immune cell profiling when compared to wild-type C57 Black 6 mice. So using flow cytometry to first assess the overall frequency of leukocytes using markers for T and B cells, NK cells, and other myeloid cells, including dendritic cells, granulocytes, and macrophages, we can see that there are no dramatic alterations in the distribution of mature leukocyte subsets in the spleen or thymus of humanized CD3E mice, as shown here in the bar graphs. To assess the frequency of T cell subpopulations, TCR beta positive T cell subsets were assessed by flow cytometry. This analysis demonstrates that CD4, CD8, and Treg cell populations, which are CD4 positive, FOXP3 positive, are present at a similar frequency to that observed in wild-type C57 Black 6 mice in both the spleen, shown on the left, and the thymus, shown on the right. To be sure that T cells are indeed functional in humanized CD3E mice, splenic cells were isolated and incubated in the presence of anti-CD3 and the anti-CD28 antibody. While wild-type mice upregulate activation markers CD25 and CD69 in response to anti-mouse CD3, humanized mice undergo T cell activation in response to the anti-human CD3E antibody, and not the murine CD3E antibody. Notably, activation studies of T cells from the humanized mice stimulated with human anti-CD3 also induced proliferation of the T cells, shown on the bottom right, at levels similar to that observed in wild-type mice stimulated with anti-murine CD3, as shown by this time course analysis. These results suggest that the T cells can respond normally to stimulation in the humanized mice. Furthermore, the humanized mice are able to secrete cytokines in response to human CD3 stimulation, as shown by the white bars, indicating an attacked CD3 signal and subsequent immune response. To ensure that humanized CD3E mice have a normal humoral immune response, ovalbumin immunization studies were performed. In the left panels, both wild-type and humanized mice are observed to produce normal anti-ova antibodies after the second and third immunization. Furthermore, a normal antibody isotype distribution is observed in the humanized mice that is comparable with wild-type C57 Black 6 mice. Finally, to assess whether T cells from humanized CD3E mice could perform tumor killing in vitro, cytotoxicity assays were performed with humanized CD3E splenocytes and murine MC38 tumor cells expressing human PD-L1. The cells were mixed at the ratios indicated and then incubated with a bispecific antibody targeting PD-L1 and CD3. As indicated by the graph, there was measurable tumor killing at both cell concentrations that increased with increasing concentrations of the bispecific antibody, further indicating that human CD3 is functional in these mice. Now that we've established the normal T cell formation and responses in this model, we next performed validation studies using tumor models to assess the efficacy of anti-human CD3 targeting in vivo. In this case study, murine MC38 tumor cells were inoculated into humanized CD3E mice, and the mice were either treated with IgG, shown by the blue line, or an anti-human CD3 antibody designed to promote T cell depletion or activation-induced cell death. As shown on the graph, mice treated with a higher dose of antibody, shown as the red-orange line, experienced a more rapid tumor growth than the lower dose in the IgG controls. This was associated with a relative enrichment of B cells and a significant depletion of T cells in the peripheral blood, as shown by the red dots on the right side. This study confirms that the CD3E mouse model is a powerful tool for in vivo and anti-CD3 human CD3 antibody efficacy studies. In these next studies summarized here, our team performed a series of bispecific antibody efficacy assessments in humanized CD3E mice. In the first panel, the tumor cell line was engineered to express human CD19, and the cells were inoculated into humanized CD3E mice and treated with blinatumomab, an antibody designed to target human CD19 as well as engage human CD3 activation. In these studies, the high doses of antibody successfully blunted tumor growth. Similar results were observed in humanized CD3 mice inoculated with tumor cells and treated with a bispecific antibody to CD3 and an undisclosed target antigen. In this third study, treatment of humanized CD3E mice bearing tumors expressing human EpCAM with a bispecific antibody targeting CD3 and EpCAM also blunted tumor growth. Altogether, these studies demonstrate that the humanized CD3E model is a powerful model for testing efficacy of bispecific antibody therapeutic candidates. In order to broaden the targeting of CD3 to include the whole complex, our team has recently generated and introduced a second-generation humanized CD3 EDG model. In this model, the entire coding region of CD3E, D, and G, as well as some of the human untranslated region, was inserted into the corresponding murine loci to replace the murine genes. mRNA expression demonstrates adequate expression of human CD3E, D, and G by RT-PCR, shown in the pink boxes. And importantly, expression of human CD3E is detected on the surface of heterozygous humanized CD3 EDG T cells by flow cytometry. Gross examination of lymphoid tissues from the humanized CD3 EDG mice did not reveal any abnormalities, and there was no significant difference in spleen weight or the cell number between C57 Black 6 and humanized CD3 EDG mice. However, the thymus weight in humanized CD3 EDG mice was slightly lower than that of the wild-type C57 Black 6 mice. Similarly, the total number of thymocytes was also very slightly lower than numbers harvested from wild-type C57 Black 6 mice, as shown on the bottom right. However, as we showed for the CD3E model, stimulated T cells from these mice appropriately proliferate in response to the anti-human CD3E antibody, as demonstrated by these CFSE staining profiles of the humanized mice, shown on the bottom right. Importantly, stimulated T cells from the humanized CD3 EDG mice were also able to secrete cytokines in response to anti-human CD3 and murine CD28 stimulation, as shown by the orange bars, similar to their wild-type counterparts stimulated with murine CD3 and CD28 antibodies, which is shown in purple. To ensure that the CD3 EDG humanized mice also have a normal humoral immune response, again, ovalbumin immunization studies were performed. In the left panels, both wild-type and humanized mice were observed to produce normal levels of anti-ova antibodies after the second and third immunization. These antibodies, again, exhibit a normal antibody isotype distribution, suggesting that these mice exhibit a normal cellular and humoral immune response. In vivo efficacy studies with our new humanized CD3 EDG model have just begun. In initial testing of the model, we again performed T-cell antibody depletion studies to determine whether blockade of anti-human CD3 could deplete T-cells and enhance tumor growth. As shown previously, on the left side, you can see that the human CD3E blocking antibodies, shown by the green line, enhanced tumor growth. And on the right side is confirmation that the therapy depleted T cells, but not B cells, as shown by the green bars in both the peripheral blood on the top and in the tumor microenvironment, as shown on the bottom. This next model will highlight the ability to test novel combination therapies in mice doubly humanized for the well-known immuno-oncology targets PD-1 and 4-1BB. In this model, the extracellular portion of both PD-1 and 4-1BB were replaced by their human counterparts as shown in pink. Importantly, expression of human PD-1 and human 4-1BB is detected on the surface of homozygous humanized T cells by flow cytometry as shown in the pink boxes. Importantly, examination of the frequency of leukocytes in the spleen and the blood of these mice revealed relatively normal distributions compared to wild-type C57 Black 6 counterparts. Because 4-1BB antibodies can exhibit some toxicity effects, it is important to ensure that these models have normal liver chemistry at baseline, and that is the case here. Next, we performed some case studies assessing the effectiveness of anti-human PD-1 antibody Keytruda and the 4-1BB antibody urelumab in preventing tumor growth of different cell lines inoculated into humanized PD-1 4-1BB mice. On the top row, the combination therapy, which is shown by the blue lines, has a significant anti-tumor effect on the colon cancer MC38 line, which is shown on the left, and the lymphoma EL4 cell line, which is shown on the right. Furthermore, when the inoculated tumor lines are engineered to express human PD-L1, you can see that the combination therapies are able to essentially halt growth of the MC38 tumors and B6F10 melanoma cell tumors, as shown on the bottom. Together, these studies highlight the powerful capability of these models to assess the effectiveness of new immune checkpoint therapies in vivo. These models are just a few models that we highlighted from a much longer list that we have available for use. So please head over to our website where you can view validation details and download our full catalog, or contact us to learn more about the models we offer. One important tool in these efficacy studies is the tumor cells that can be engineered to express human antigens. So here is the current list of humanized tumor cell lines that we offer. In addition to these off-the-shelf lines, our gene editing team can also generate humanized tumor cell lines if the cell line is provided to us. To summarize this part of the talk, I hope you can appreciate the translational challenges in discovering new immunotherapies and how our multiple strains of humanized mice can help to improve translation of future drug candidates for immuno-oncology. We validate the expression of humanized proteins, immune cell frequency and maturation, and do our best to perform functional validation using reference antibodies whenever available. Thus, our humanized mice allow for robust preclinical modeling and testing for novel immuno-oncology drugs, including mono and bispecific or multispecific antibody candidates, novel combination therapies, and preliminary toxicity assessments. Using these robust humanized models we have also been able to successfully advance our own product pipeline. In particular, our CD40 antibody in our pipeline showed excellent safety and efficacy in our CD40 humanized mice and has just been approved for phase 2 studies. If you're interested in learning more or interested in partnership opportunities, please contact us. For the final few slides, I would like to introduce our humanized immunoglobulin models that can help further support your therapeutic antibody discovery applications. These models, RenMab and RenLite, express the entire human heavy chain variable region in place of the murine counterpart. Now, while RenMab mice carry the full kappa light chain repertoire, our common light chain, or RenLite mouse platform, is specially designed for advancing bispecific and multispecific antibody discovery because it solves the common issues of the bispecific antibody discovery process, which are immunogenicity and chain mispairing. So the immunogenicity is reduced because of natural in vivo selection and affinity maturation of the human heavy chain repertoire. At the same time, it allows for efficient construction of different heavy chain variable domains that share the same light chain for future bispecific, multispecific antibody engineering at later stages of development, and which ultimately saves time and effort. Because we have a very strong gene editing technology platform, we've also launched an initiative to further engineer these Ren mice. So we refer to the second generation as the Ren mice HITS platform, which consists of a library of Ren mice, each with a specific target gene knocked out. So why do we need to perform antibody discovery in a knockout mouse? So when generating antibodies, one common issue is the high homology between human and mouse. So in this case, when we knock out the target gene in the mouse, the immune tolerance is broken, and a much stronger immune response can be elicited. This same strategy can also be used for challenging targets, including GPCRs or ion channels. Our RenMab HITS mice can also generate antibodies that bind to broader epitopes for a specific target. Additionally, it can be very useful to generate antibody candidates that react to both human and other species used for preclinical studies and validations. Therefore, there will be no need to make a surrogate antibody for a preclinical study validation. On our RenMab website, you can see the status overview of the knockout mouse generation. So right now, we have nearly 700 targets that have been knocked out in Ren mice. So once the HITS mice are homozygous, you can see on this timetable that we move forward with immunization, followed by antibody discovery, in vitro, and finally in vivo development. And the number of mice at each stage is obviously listed here in this screenshot. So among these 700 targets, 11 have been exclusively licensed out to partner companies. So on the website at renmab.com, you can search for your target of interest and contact us if you are interested in partnership. Given the powerful potential of our humanized RenMab and RenLite mice, we offer in-house antibody discovery services for HIT generation with a platform focused on in vivo antibody generation. So to give just a very brief overview, we immunize and identify mice with strong titers, and their B cells can then be isolated and enriched before loading onto our Beacon optofluidic system for single B cell sorting. Within just one day, all the antigen-specific B cells can be identified and sorted out. These positive B cells can then be lysed and their mRNA extracted, followed by single B cell sequencing. These sequences can then be cloned into expression vectors and are validated by quick transfection and ELISA. So altogether, this entire platform can take you from immunization to purified recombinant monoclonal antibodies in just 10 to 13 weeks. So there are a few main advantages of this antibody discovery workflow. First, the Beacon optofluidic system significantly shortens the discovery turnaround time from months to weeks. Second, the positive antibody sequences in this platform are obtained at a very early stage to avoid loss of positive clones during lengthy subcloning or selection procedures. And finally, the B cells that we capture have already undergone affinity maturation in vivo. Therefore, the antibodies derived from this workflow typically have very high antigen binding affinity, with many in the subnanomolar range. With that, I hope you can appreciate how our integrated technology platforms at Biocytogen ultimately work together to streamline, support, and accelerate the discovery of novel antibody therapeutics, including bispecific and multispecific antibodies. So we hope you'll further explore our services, and we look forward to working with you in the future. Thank you very much for your time today, and please feel free to reach out with any questions you may have.